Yes, sir. Joined. Good morning, sir.
Sir, on behalf of our organizing committee of National Workshop, I welcome to our chief guest, Dr. GBK Prasad sir, our Honorable Pro Chancellor sir, Sri Lieutenant General VK Sharma sir, Aviation retired. I also welcome to Honorable Pro Vice Chancellor Research sir, Professor MP Kaushik sir. I also welcome to officiating Vice Chancellor sir, Professor Dr. Anil Vasis sir, and all the participants.
Yes, sir. Education requires costly resources like time and money, which are often hard to find. But those who do invest these resources often end up richer, not just because they often improve their financial situation, but because their minds are richer too. A very good morning and warm welcome to our chief guest, Professor Dr. GBKS Prasad, Jivaji University, Gwalior, Honorable Pro Chancellor, Lieutenant General VK Sharma, Ati Visis Seva Medal, officiating Vice Chancellor and Pro Vice Chancellor, Professor Dr. Anil Vashist, Pro Vice Chancellor Research, Professor Dr. MP Kaushik, Professor Dr. Vikas Srivastava, officiating Head, Amity Institute of Biotechnology, Amity University, Madhya Pradesh, Dean Academics, HOIs, HODs, faculty members, participants, and students of Amity Institute of Biotechnology, Amity University, Madhya Pradesh. I, Dr. Neha Sharma, Assistant Professor, AIB, feel privileged to welcome you all on behalf of Amity Institute of Biotechnology, Amity University, Madhya Pradesh, to the 4th National Workshop cum Hands-on Training on Advanced Techniques in Molecular Biology, Medical Biotechnology, Industrial Microbiology, and Bioinformatics. Today, we all are gathered here as wisdom comes not from age, but from education and learning. As a genius said, the aim of education is the knowledge, not of fact, but of values. It is an occasion in which we get opportunities to test our knowledge and understanding. We look forward to get an exposure about what the best of brains think about emerging techniques. To make this event a blessed one, we invoke Goddess Saraswati by Saraswati Vandana for seeking the choicest blessing.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः.
Thank you, everyone. Thank you, ma'am. So, the guiding force behind our constant efforts is Honorable Pro Chancellor Lieutenant General VK Sharma. I feel immense pleasure to introduce you Honorable Pro Chancellor Lieutenant General VK Sharma Ati Vishish Seva Medal. Dr. Sharma is an alumnus of erstwhile King George School, Shimla. He did B.Tech from College of Ministry Engineering, Pune, M.Tech degree from Pune University, MMS from Osmania University, all in first class with honor. He joined the National Defense Academy and passed out as a permanent commissioned army officer from Indian Military Academy in 1972. He was granted the President Commissions of the Corps of Signal in the Indian Army. He served the Indian Army for almost 40 years and retired from the second highest rank of Lieutenant General, which is the 3-star and the flag rank of the Army. He participated in the opening Operation Rakshak, Operation Vijaya during the Kargil War. He also headed military delegation to France, Germany, and USA. For his meritorious service, he was awarded Ati Vishish Seva Medal by Honorable President of India on January 26, 2020.
2011. General Sharma is an excellent academician too. Under his able guidance, Amity University, Madhya Pradesh has grown by leaps and bound towards excellence, and he has been awarded as the best private university of Madhya Pradesh. May I now request Honorable Pro-Chancellor Sir Lieutenant General V.K. Sharma for his opening remark.
Thank you very much for the introduction. Professor Dr. G.B.K. Prasad, who is the expert lecturer for today in the inaugural session. Dr. Vikas Srivastava, the officiating HOI of Amity Institute of Biotechnology. Dr. Anil Vishish, the officiating Vice Chancellor. Professor Dr. M.P. Kaushik, the Pro-Vice Chancellor, Research. All the others who are present, and my dear students. The topic for today's national workshop cum hands-on training on advanced techniques in molecular biology, medicinal biotechnology, industrial microbiology, and informatics is very relevant to all of us because it affects all humanity. Today, with increase in the wars which are taking place, leading to various decapitations, the pollutants resulting thereof, and the global warming, which is happening across the world. The humanity is heading towards extinction, as has been predicted that in all evolutions which become highly technical, it can happen that the advancement and the technology destroys that race itself which created such technology. I dare to say that some of the astrophysicists have written various books on that there have been maybe other extraterrestrial bodies where life has come up, and then because of very high state of advancement that they have achieved, that has resulted in the destruction of the race itself. That may not happen to us, provided we take measures now to stop all this, what is happening. And this can be done through biotechnology, which has been shown that during the pandemic, it was the biotechnologists, the biologists, the scientists working at molecular and atomic level, the scientists working on genome, scientists who work day and night to produce the vaccine, which saved the mankind from further losses that could have taken place. The pandemic was controlled because of this category of people and scientists and hard workers that I just mentioned. The informatics is one branch which is important because it collects all the data. The development of modern science in medicine requires the collection, integration, interpretation of molecular, genomic, and cellular data together with clinical data. That is most important. Therefore, it produces a significant amount of challenges to bioinformatics. And for this, the investigations and understanding of biological complexity, a variety of methods and softwares have been created and developed and are still developing. To speed up biotechnological developments, bioinformatics technologies such as sequence analysis, matching, of molecular modeling, docking, indexing, and simulation techniques are used. Numerous upcoming bioinformatic breakthroughs are predicted to promote the study of large amounts of biomedical information, and consequently, bioinformatics will play a critical role in analyzing different types of data created by high throughput research methods such as genomics, transcriptomics, and proteomic data sets and databases, and then arranging the knowledge gathered from traditional biomedicine. We must remember our traditional system of medicine should not be lost track of, because so far we have maintaining very good health in India and have overcome all these diseases through our traditional systems. So the traditional systems should not be junked, they should not be forgotten, but they should be rather taken as a base from where we should build the new technology and integrate the two in such a manner so that the mankind derives maximum benefits from all this. Another important thing is that while the vaccines are being done and the research is being carried out primarily on animals first and then on humans, but in view of the ethics involved, the dependence of research on animals will have to be reduced. Today, as our human mind becomes more developed, we realize the pain of inflicting pain on animals which also have got life, and we do not wish to make them suffer in the manner that we used to do when our own minds were not that much evolved and the humans were behaving like animals themselves. With that, I wish this workshop, which is going to last for about 8 days, a great success. And those who are participating in the workshop, please remember the difference between a workshop and a webinar or a seminar is that in the workshop, you get enough time to ask questions, to do hands-on training, and to gain useful experience that can be used for the rest of your lives. And the expert advice, which is available from the experts who are always present and willing to give knowledge. Knowledge sharing has been our tradition in India, and we do not never believe in hiding the knowledge or not giving it to people. Anybody who deserves to get knowledge is given knowledge. That is what our tradition and our culture says. So, my dear students and the participants, please prepare yourself well, take advantage of the situation, and make sure that the workshop is a success and you gain by this. This is the 4th workshop that MIT University, Madhya Pradesh is organizing on this topic, and every year we have been doing it for last 4 years, and we hope and carry this tradition forward for remaining times to come. My best wishes to all of you. Gain knowledge, stay updated always, and contribute towards the health of the nation and the world through technology, through the Indian knowledge system, and through furthering the research in this direction. Thank you very much, and Jai Hind.
Thank you very much, sir. Your thought-provoking address set a perfect platform for our speakers and participants to deliver their presentations in their area. Now I feel delighted to introduce our chief guest and today's guest speaker, Professor Dr. GBKS Prasad. Dr. GBKS Prasad, Professor of Biochemistry, Jivaji University, Gwalior. He did PhD in 1998. He received various fellowships and visited outside India on various academic assignments like Commonwealth Fellowship for Postdoctoral Research at Liverpool School of Tropical Medicine, Liverpool, UK, Texas University, USA under Indo-US Collaboration in Science and Technology Program, Imperial College of Science and Technology, London, and World Health Organization, Geneva. Dr. Prasad have about more than 30 years of teaching and research experience in the field of diagnostic and biomedical research, including anti-diabetic herbal and Ayurvedic formulation, and is currently leading a good research team. He got about 10 research projects funded by various public and private agencies on various aspects of diabetes mellitus. He authored two textbooks, that is, Molecular Diagnosis, Promises and Possibilities, published in 2010 by Springer, USA, and Microbes, Concept and Application, published by Wiley Blackwell in 2012. He has published more than 120 research papers, various reputed international and national journals, and 7 patents has been filed. He supervised about 20 PhD students and guided more than 100 MSc dissertations. He is member of various international and national scientific bodies and reviewer and editor of national and international journals. May I now request Professor Dr. G.B.K. Prasad to kindly deliver the expert lecture. Please, sir.
Thank you. Thank you very much. My regards to Professor Sharma, our Pro-Chancellor of MIT University, Acting Vice Chancellor, and Kaushik Sir, Pro Vice Chancellor R&D, Professor Vikas Srivastava and his whole group for organizing such a workshop in sequence for the last 4 years. Generally, the universities and the higher education institutions, their responsibilities can be divided in under two categories. One is the creation of energy and dissemination of the energy. And creation of energy, that is through the research, and dissemination of the theoretical knowledge as well as the practical knowledge to the students. These are the two main responsibilities of any higher educational institution. And as far as the biotechnology is concerned, as mentioned in the introductory remarks of our Pro Vice Chancellor, it is a multidisciplinary subject, and it is there, it takes inputs from each and every branch of science, starting from physics, chemistry, and biological sciences, botany, animal sciences, environmental sciences, microbiology, agriculture, and whatnot. Microbiology, of course, that is one of the key subjects playing a very important role in biotechnology. And that way, one has to have the basic knowledge and concepts of all branches of science before picking up or before taking admission in biotechnology. And leave aside the engineering programs in biotechnology, that is B.Tech. But generally, we advocate at graduation level, that is at the conventional BSc level, and biotechnology should not be offered. And because unless the student is exposed to different branches of the biological sciences, and unless the basic principles of various disciplines are clear, they cannot understand the the intricacies and the cannot appreciate the biotechnology. Because what is biotechnology? Biotechnology is nothing but the fusion of biology with technology in simple terms. So whatever developments that have taken place in recent decades, they are being exploited in almost every branch of science, and that would result in generation of some or other, you can say, ultimately a constructive outcome, or in terms of some product, I can say. So that way, every program, in fact, at the post-graduation level, if not at undergraduation level, and undergraduation level, if it is an honors program, 4 years program, and there also one should have a, a dissertation. It's a very compulsory dissertation should be there, and where the student is assigned to work on a given problem or in a given area, and allow uh, the the innovative ideas of the student, and let them design one research problem and work on it over a period of 6 months to 12 months time, at least minimum, and that should come out, that should result in the generation of something, a kind of, you can say, a product or a prototype product, which can be refined further, and it can be, uh, you know, it can be made commercially viable. So that way, almost, one of the programs, the basic flaw of biotechnology programs in our Indian system or universities of different higher organizations is that, and biotechnology students are also taught mainly the theoretical knowledge like any other branch of science, and they are not, trained uh, in development of a kind of product. Means, it is a, you name any discipline, biotechnology is there, whether it is a plant sciences, plant tissue culture is there, and genetically engineered plants are there, uh, like BT cotton, for example, one is very popular among the public also. What is BT cotton, just if we talk about it? And the plant which is genetically engineered develop resistance to various pests. So what is done is that there, for example, and there are some bacteria, uh, particularly here in this case, BT cotton, Bacillus thuringiensis. It has uh, it has the ability to synthesize and secrete some toxins which are toxic to different, the nematodes and some insects also, like even they have got mosquito larvicidal property, and that particular gene is isolated and it is inserted into the plant genome, and we say that yes, now it is a genetically engineered plant, and it can synthesize toxins so that it develops resistance automatically against the various pests. And also, if we talk about the agriculture, for example, the same way, there are fertilizers are there, biofertilizers are there. So what are biofertilizers? They are nothing but the microorganisms, and they make the soil nutritionally enriched. And just to give you an example, here a microorganism means not necessarily it has to be a bacterium. It can be a fungus also, mycorrhiza. So there are two types of mycorrhiza: ectomycorrhiza and endomycorrhiza. So their main function is to uh, accumulate the phosphorus, gather phosphorus available in the soil, and get accumulated, make it soluble, and make it available to the plant. And of course, everybody knows the role of rhizobium, the fixation of the atmospheric nitrogen. So that way the microbes play a very important role in majority of the biotechnological, the processes in various you can say the living things, including plants, for example, animals, of course, innumerable applications of biotechnology. There are several sub branches of animal biotechnology, as mentioned, in the initial remarks by our Pro Vice Chancellor during this COVID pandemic. The molecular biology, the techniques of molecular biology played a very significant role in the diagnosis, particularly RT-PCR. It is known to even a common person also. So there, the molecular biology has contributed not only in genetic engineering, it has been exploited for diagnostic purposes also. And the same molecular biology can be exploited, means the molecular biology is nothing but the study of the genes and their arrangement, and then what are the processes by which they get expressed, and what are the intricacies involved in the transcriptions and translation processes and all those things. So they can be exploited for therapeutic purposes also in case of medicine. And another important thing is that, the vaccinations in medical biotechnology, if we come to that one is diagnosis, and second is therapeutics. And diagnosis also has got, the biotechnology has played a very important role in improving and in refining the diagnostic approaches, and that I'll be discussing in detail later on. And also, when we talk about uh, the conventional biotechnology, or you can say classical biotechnology, because it is not biotechnology, although the name has been coined a few decades ago, and I think it was in 1919, it was coined by a German scientist. It has been there. The biotechnological processes are in, are regularly being used for centuries together. Preparation of the curd, preparation of alcoholic beverages by fermentation. They are nothing but the classical biotechnology only. Their microorganisms are playing a very important role also. The only thing is that now the recent technologies, so now they are refining these biotechnological processes, and one important area is, you can say, the nano technology. And this nanotechnology is also being exploited by the biological systems for development of, you can say, nanomedicine. Again, nanomedicine, it includes the diagnosis as well as the uh, therapeutic approaches are there. So that way, I'm very happy that the MET has been organizing regularly, on annual basis, the hands-on practical training in development of various, in demonstration and hands-on training in various molecular biology techniques which have got relevance, uh, particularly in the diagnostic area. And also, of course, as mentioned by this one in the bioinformatics. Bioinformatics is one thing. Generally, it has several ramifications are there. And when we go for generally, research, and we start with the in silico approach, that is the docking studies, as mentioned, and drug targeting. Just instead of fishing out, when you have to work out a drug for a particular disease, and which is a new disease altogether and nothing is known, so we can use these bioinformatic tools for identifying the drug target and drug candidates, and they can be again extended to the wet laboratory using different either plant system or the animal system or microbial system. So that way, so, and the students are going to be exposed to all these different softwares which are commonly being used um, in molecular biology during this training program. I am very happy and I congratulate the whole team of the Institute of Biotechnology, Professor Vikas Srivastava, and I think the program has been initiated by Dr. Rajesh Tomar when he was there, and that is being continued. And I'm sure the students should make best use of the training program and try to develop expertise, if not in all techniques, in certain techniques by the time the workshop is over within a week period. So that is what I wanted to convey now. And should I go ahead with the lecture also now, Neha?
Yes, sir. Yes, sir.
Okay, let me share my slides.
Okay, sir.
So, what is the role of biotechnology in medicine? As I mentioned,
Sorry to disturb
in
you,
my
sir.
initial,
Kindly make it full slide show.
yeah. Okay.
Sir, not yet.
Not okay. 1 minute. Slideshow is there, and we make it full form. Full, it is not going. Is
Sir,
it full form?
at the right corner, sir, there is a that screen symbol is there. You just click it on that. The three icons are there, out of which one is the full screen. At the right
Otherwise,
corner.
F5, you can go by F5.
F5 can also.
F5. Okay.
Oh, let him continue. I think.
Okay, sir, you continue, sir.
कहाँ है देखो नहीं राइट हैंड कॉर्नर में 3 बोलता है ना.
So, Dr. Prasad, you can please continue like this only. Don't worry. There are, we can see that.
Yeah. Okay.
Yeah.
Right. Just let me talk in simple terms. When we go to any physician with some complaint, generally what we do is that we give our the history, clinical history to the physician, that whatever symptoms that are there. Uh, we present them that I am suffering from some fever or some any cough or cold or any other problem, breathing problem and everything. And on the basis of that one, the doctor would arrive at some diagnosis. That is what we call clinical diagnosis. And because of the overlapping of symptoms by different diseases, could be possible that the clinical diagnosis alone may not help in arriving at a correct diagnosis. So then the correct diagnosis is made by the laboratory investigations. That is what we call it, laboratory diagnosis. Sorry, just yeah, ठीक है. नहीं हो रहा है बताना, उनका नहीं हो रहा है. Next, the clinical diagnosis is to be confirmed by the laboratory diagnosis. What are different types of laboratory diagnosis? Like microbial diagnosis means, depending on the season and the symptoms, the doctor might suspect that the person is suffering from some infection, a bacterial infection or a viral infection. Then he will ask us, the physician would ask us to get some investigations done from microbiology laboratory. So it may include the collection of the samples, and then also if it is if any bacteria is found, maybe depending on, as I said, the season and the prevalence of different types of diseases. So whether it is a typhoid or a cholera or any other bacterial infection or a viral infection, the physician would direct us to a microbiology laboratory. And if it is a bacterial test, generally the doctor is expected to ask us to go for antibiotic sensitivity test also, just to ensure that yes, appropriate antibiotic is prescribed to the patient. But generally what is done in practice is that rarely any doctor would ask us to go for antibiotic sensitivity.
Dr.
And
Prashad, please change your slide. The next, you're talking on the next slide. You change the slides which you are showing the laboratory diagnosis.
yeah,
Where they all...
I'm explaining that slide only, sir.
Okay. No, but that's not visible to us.
Actually, it is not visible. Okay. Oh, इसमें मत जाए मेरे ख्याल से.
It's not moving.
Okay, sir. Okay.
Sir, kindly share it again. Maybe it is hang, sir.
शेयर करो दोबारा.
That's fine. Now it has moved.
Yeah, right. Thank you. Yeah. So that is in microbiology diagnosis. That is what we call it. Then pathological diagnosis. Suppose if doctor suspects any kind of, let us say, any kind of pathological condition that is like a cancer, don't show that. That it is not moving. Let it be on that. End the show. End show. Yeah. Let it be like that. So in a pathological diagnosis, generally the histological sections are taken and then they are stained by different conventional histological stains, maybe fluorescent stains also, and then accordingly the diagnosis is provided. And in biochemical diagnosis, particularly if these diseases are mainly they are not diseases, but they are disorders of the lifestyle disorders like diabetes, or any kind of liver problems or kidney problem or any organic disease, generally the physician would refer us to the clinical biochemistry laboratory. And then of course depending on the symptoms, the physician may ask us to go for the X-ray or CT scan or MRI or anything like that. That is what we call it, the radio diagnosis. And then immunological diagnosis, particularly immunological and molecular diagnosis. These two are considered to be the advanced ones. And for diagnosis of accurate diagnosis of the various infectious agents, including different microorganisms, and either by detecting the antigens that are released by the pathogens, the antigen detection tests are developed, what we call it enzyme immunoassays. And the second is the molecular diagnosis, because here this is molecular diagnosis. Molecular diagnosis is much more sensitive than the immunological diagnosis also, by identifying a small patch of DNA which is present only in that particular infectious agent. So that way means different laboratory diagnosis are available, and usually the physician would confirm uh, the diagnosis only after getting the report from the referred laboratory, and it is done. So everywhere, in every laboratory, biotechnology is involved. For example, in microbial diagnosis also. So there also nowadays, what is being done is that instead of the conventional microbial identification, so the microbiology laboratories are using the immunological approaches for detection of a given pathogen, or a molecular approaches for identification of a given pathogen. So they are nothing but the biotechnological tools only. And in pathological diagnosis also, in cancer, particularly the markers which are being used, generally, of course, the monoclonal antibodies against a particular tumor specific antigen is used, and these antibodies are conjugated to some either the fluorescent dyes or to enzymes also. Peroxidase is very commonly used, and they are nothing but the biotechnological tools. And in biochemical diagnosis also, particularly in majority of the cases nowadays, the biotechnological approaches are used for diagnosis. For example, if hormones are to be detected, that is a very common problem. And there, again, the immunological approaches are used for detection of a given biochemical marker from the fluid. And radio diagnosis, of course, a lot of developments have been made, and depending on the nature of the problem, if it is associated with the lungs, they ask us to go for this one, or any kind of abnormality in the body at anywhere from top to bottom. So the physicians would advise us to go for the the scanning, a CT scan or the MRI. These are the two advanced techniques which are commonly used. Or if one particular organ is to be scanned, preferably, again, there are methods and different the tools are there where only that particular organ is scanned in a in a detailed way, and that way all laboratory diagnostics they involve biotechnological tools. So now here my focus will be on the role of nanotechnology in medicine. Can you see this slide? Is slide visible?
No, not yet.
Yeah.
Not yet, sir.
Sir, slide is visible but not moving.
Acha, okay.
Sir, I think you need to unshare and then reshare.
Not more.
Now
It's
it is
okay. It has
moving,
come
sir.
now.
Now it has come.
Yeah, right,
It's a little bit slow, I think.
right. So generally, what is nanoparticle? Nanoparticle generally any particle with a size in the size range of 10 nanometers to 100 nanometers is considered as a nanoparticle, and a smaller the particle and the utility is more, and there are some particles like quantum dots. They have the size less than 10 nanometers. They have again wider applications in all you can say three categories: in diagnosis, as well as in immunoprophylaxis, prophylactic methods, development of prophylactic methods. Third is in therapy also. So that is for early detection and imaging diagnosis, that is radio diagnosis, and therapeutics also. So quantum dots. So quantum dots, of course, although they are not very popular nowadays, and lot of it is one of the hot areas of research to make use of these quantum dots for biological purposes. So as I mentioned that these quantum dots, they are quantum dots are nothing but semiconductor nanoparticles, and mainly they are, you can say, if you see the nature of these quantum dots, particularly the cadmium and tellurium are used there, and one important thing is that their size is generally 10 nanometers, and they are hydrophobic in nature. They can be made hydrophilic by coating different ligands, and we can conjugate proteins also with the quantum dots, and that way means the antibodies can be conjugated to the quantum dots, and these quantum dots will help in you can say drug deliveries, as well as the drug targeting, particularly in cancer, means they are being experimented. uh, Majority of the cancer drugs are toxic in nature, toxic to normal cells also. So cancer is such a disorder where long-term treatment is required. And when we give conventionally through intravenous mode or any other mode, usually the therapeutic agent is distributed throughout the body to all tissues and the cells also, and normal cells they get damaged when they are exposed to these things, and um, if can develop a target specific delivery, so the drug
I think sir is disconnected. Vikas sir, Prasad sir is disconnected actually.
Sir, I am talking with him.
He is joining, sir.
He is joining, sir.
Yes, sir has
Actually,
joined.
some issue was there. So he told me that I'm joining in 2 minutes.
Very sorry, sir, for repeated interruptions from my side. And I hope, I think hereafter there should not be any kind of interruptions.
No
But
issues, sir.
anyhow, the quantum dots are being exploited. One is for diagnostic purposes, and another is for drug delivery and drug targeting. So that way they are under experimental conditions, but I'm confident that in another 1 year, the quantum dots will have a significant role in medicine. I'm skipping these things. Right. And here I want to just, the present what we have been doing in the field of nanomedicine. Particularly, our focus is on the therapeutics, exploitation of the nanotechnology for therapeutic medicines. And we have prepared nanoparticles of, nearly about 10 different herbal components. And these nano formulations, that nanotherapeutic compounds, so they have been validated, and some of them are being validated in vitro systems, and also in vivo animal models, and some of them we have now extended to the human beings also. Take, for example, curcumin. A lot of studies have been done, and nanoparticles are available and made by different people also. The only thing is that we have simplified the process of nanoparticle preparation, that I will show, because this being the workshop, I thought that let me show the simple steps which are involved in preparation of the nanoparticles, and I'll show you how they have been prepared, and what are their functions. And these are different other nanoparticles which are used, and they are being uh, uh, experimented in diabetic animals. And actually, when we question ourselves, when it is a nanoparticle, what is the need for a nanoparticles in case of curcumin? Curcumin, it is in fact, many of you might be knowing that it is an active compound present in the turmeric, haldi, and the main bottleneck is that curcumin concentration is only about 3%. That means if we take 100 grams of haldi, only about 3 grams is the active compound which will enter our bloodstream also. and it is hydrophobic nature, and the absorption is not 100%. And the bioavailability also is found to be very low. So just to overcome these two problems, we thought that whether nanoparticle preparation, and to a extent we have we could prepare the nanoparticles by simple approaches, and we have shown that the nanoparticles is very high compared to the curcumin. Also, they have a very good therapeutic application. It is the nanoparticle preparation. If you see here, there are only very simple and few steps which we have made, and also. And this is
Sir, your slides are not visible. Yeah. Now it is visible.
right. Yeah, there are different steps, whatever I have been explaining. Mainly, it is two important things: homogenizer and sonication and centrifuge. These are three simple machines that are required for preparation of the nanoparticles, and some manipulation at one or other step is needed for preparation of nanoparticles from other herbal compounds or from other herbal sources. And thereafter, just to improve the drug delivery, we have conjugated this nanoparticle with a chitosan. It is nothing but a inert carbohydrate, and then the absorption rate is found to be very high when it is conjugated to the chitosan. And again, that is also very simple. You make nanoparticles of chitosan and nanoparticles of curcumin, and simply when you mix them, automatically formation chitosan nanoparticle are generated. And thereafter it is subjected to characterization by various biophysical techniques, and of course, first thing is that assess the size of by using particle size analyzer, and if the size is less than 100 nanometers, and they can be used as it is, and if it is more, then some standardization is required. Then it is characterized by the scanning electron microscopy, transmission electron microscopy, atomic force microscopy, XRD, DSC, FTIR, and zeta potential. So these are all techniques, biophysical techniques, which are used commonly to characterize the nature of the nanoparticles. All those things are done. So I am skipping all those the images. And then what we have done is that after that we have administered this curcumin. Just we wanted to find out this curcumin has got a number of functions, because it is conventionally we know it is a very antibacterial compound, and not only this one, it is anti-cancerous. You name any disease, curcumin has that potential. And our interest was to this curcumin can help Hello.
Yes, sir. Your voice is coming, but slides are not visible, sir.
You can switch off your camera, sir. Actually, your bandwidth is low.
इसको reshare करें। क्यों ये प्रॉब्लम बहुत दे रहा है इसे?
Now it is visible, sir.
Are they visible, sir, now?
Yes, sir. It is
Yeah,
visible.
right. Yeah, right. I'm not going into the all details. Just yes, the curcumin has been shown to be anti-hyperglycemic agent, and we wanted to identify what is the mechanism that is involved in bringing down the glucose levels, whether it is by increasing the secretion of the insulin, or it is by promoting the by reducing the insulin resistance, that is by promoting the function of the insulin, or by manipulating some of the adapter molecules, so in this way. Um, uh, uh, so that way we have identified, and actually this curcumin, it works at the cellular target cell level, and there is a GLUT4 translocation, because this is
Sir, you can switch off your camera and then share the PPT. Hopefully, the problem will not come.
okay. Can you see the slides now?
No, sir, not yet.
Sir, it's about to visible. It is.
But what we found out is that it helps in the translocation of GLUT4 to the plasma membrane, which is very much required. A transporter protein which transfers, because glucose to be utilized for generation of ATP and everything. From the blood, the glucose is to enter cell. At that point,
Sorry to all dignitaries and participants. Sir is facing some network issue. Vikas sir, kindly call to Prashad sir.
I am on phone. Sir is connecting in few seconds.
So next time ठीक है, अगर करेंगे तो ठीक है. And then we have studied the antioxidant, anti-inflammatory functions of curcumin, because you name any disease that is and the complexity arises with the building up of the oxidative stress and the inflammatory inflammation, and this curcumin helps in reducing the inflammatory response as well as the oxidative stress also. And at the DNA level also it functions, and generally conventionally the comet assay is done to see to prevent the whether the curcumin is working or not. That is one thing. And thereafter, of course, what we did is that again, this is I don't know whether this slide is visible or not. This is one study we have done in association with MIT University. Dr. Vikas Srivastav, he is associated with this project, that is preparation of the zinc nanoparticles, curcumin zinc nanoparticles, and a ointment also has been prepared by them, and I think it is patented also. And this ointment, it has shown very good healing. I think this is composite ointment because I think it is okay.
Your voice is breaking, sir.
Just these are the slides which I am showing, that has been conducted in association,
Yes, sir. Yes, sir. Yes,
with
yes,
our
sir.
laboratory.
Yes, sir. Yes, sir.
By your group, that is
Yes,
zinc
sir.
oxide nanoparticles and the silver nanoparticles. Green synthesis of these nanoparticles has been done. Yeah. So, and again, these ointments have been studied in experimental models also. That is, wounds are generated, excision wounds are generated in animals, and it's been shown that if you compare with the curcumin, this reference one is the betadine, is used as a topical ointment. Used in hospitals now. This is coupled with the zinc CSC, that is chitosan. So if you can see that one, the curcumin coupled to zinc and chitosan. So that is a wound healing property compared to the but anyhow, I am very sorry for all repeated interruptions for one or other reason at my end. And what I wanted to just inform the students is that nanotechnology, application of nanotechnology in biology is one of the priority areas. And I think in the coming 1 or 2 decades, coupled with the artificial intelligence, definitely this nanotechnology or nanomedicine is going to rule the whole, you can say, the किसी का फिल्टर नहीं आ रहा. Yeah, I am done. But I am very sorry. I think because of so many, the lapses from my side, whether the message is conveyed properly or not. Thank you very much.
Thank you very much, sir.
Am I audible to you, Neha?
Yes, sir.
Yes, sir.
Am I
Yes, sir. You
audible?
are audible. Yes, sir. You are audible,
Right.
sir.
Right.
Thank you very much, sir, for enlightening us in the area of biotechnology like nanobiotechnology, medical microbiology, immunology, and other fields. It was very informative and helpful for us, our participants, and students. Thank you once again, sir. Now I would like to request all the dignitaries to kindly release lab manual. Thank you all.
Thank you, everyone. May I now request Professor Dr. Vikas Srivastav, officiating head, MIT Institute of Biotechnology, MIT University, Madhya Pradesh, Gwalior, and the co-convener of the workshop to kindly give vote of thanks. Professor Dr. Vikas Srivastav obtained his PhD in biochemistry from Cancer Hospital and Research Institute, Jiva University, Gwalior. He has more than 21-year teaching and research experience. He has published more than 50 research papers in various journals of international and national repute. He has been granted one patent from the Patent Office, Government of India, and published the five patents in the Patent Journal of India. In addition, he has filed a two patent in the Indian Patent Office. Two research projects funded by the Madhya Pradesh Council of Science and Technology, Bhopal, are being run by him as a principal investigator and the co-principal investigator. He has also published two books in the form of lab manual and one book chapter. He has co-supervised two PhD scholars for the award of doctorate degree in the biotechnology and currently supervising 4 PhD scholars. He has presented the paper in various international and national conferences. Presently, Professor Srivastav is involved in the designing and synthesis of multifunctional nanomaterial by chemical and biological route and the clinical biochemistry. He is a member of various international and national scientific bodies. Please, sir.
Thank you, Dr. Asha. It is my privilege that today I got an opportunity to propose a vote of thanks on the occasion of inaugural session of 4th National Workshop cum Hands-on Training. On behalf of organizing committee, first of all, I would like to thank to our chief guest, Professor GBKS Prasad, for accepting our invitation and enlighten us by his experiences and thoughts. I am also thankful to our guiding force, Honorable Pro-Chancellor, Lieutenant General V.K. Sharma sir, for his constant support and guidance at every stage. I am also thankful to our Vice Chancellor sir, Professor Dr. Anil Vasist sir, for constant motivation and support for this event. I am also thankful to Pro Vice Chancellor sir, Professor Dr. M.P. Kaushik, for his kind suggestions and guidance at every moment. I am also thankful to all the HOIs, HODs, Dean, Registrar for gracing the occasion. I am also thankful to the administration team, Mr. Colonel Sethi sir, For logistic support, Brigadier Gangele sir. IT team from Gwalior as well as Noida, who has constantly supported us for the event. And ASCO team, who has provided the support for the wide publicity. All the participants from different institutions who has joined us in an offline mode as well as online mode for their training and hands-on training and workshop. The team AIB also. So I am thankful for everyone, whoever is associated with the program. Thank you very much, sir.
I would like
Thank
to
you, sir.
convey my personal thanks to.
Now I request to
Just
all,
1 minute.
Dr. Neha,
kindly
please.
open your camera.
Dr. Neha, just
Neha,
1 minute.
please.
Listen to me. I would like to convey my personal thanks to Dr. Prasad, who has been always supporting Amity University during all various functions and various events. And sir, your guiding force, especially towards biotechnology and as a subject matter expert also, you have been kindly helping us. So my sincere thanks and gratitude to you.
Thank you, sir. Thank you, all the dignitaries. Thank you.
Thank you.
Thank you, sir. I request to all, kindly open your camera for group photo. Thank you, sir. Thank you all.
Thank you, sir.
Thank you.
Thank you, all the dignitaries.
Now I would like to
Thank you very
request
much.
participants to move for hands-on training session by joining the session through link provided, and those are presented in offline mode, please assemble in Biotech Lab for first session. Thank you, sir.
Thank you.
Thank you, sir.
Can I close the session, sir?
You can close the session.